up and formed into vacuoles. So you can't see that because the plasma membrane is way too small. But what you will see is those red-colored yeast be ingested, and they'll be a part of this vacuole. And then the lysosome would fuse with it to digest it to get nutrients, okay? You won't see the lysosome, but you can see those food vacuoles, okay? So the permeases are rather large. They move rather quickly. To slow them down, add some methocel. It's a very viscous solution. It's like you trying to swim in honey. You're not going to swim as fast, okay? That's how it works. It doesn't anesthetize them or like that. It just slows them down. Here's an example here of Nightella. This is a cell here. So you can see the scale. That's 1 centimeter. This one cell here might be 6 centimeters. That's huge. As a result, you can't rely upon free diffusion to go from there to there. It's just too long, the cell would die. But if you do the bulk mixing, you don't have that problem. And you see the bulk mixing because you will see the chloroplasts, again, because they're green, high contrast, you'll see the chloroplasts move around throughout there, okay? And there's specific things that help that, cytoskeletal. Here's Euglena, again, scale bar. So that red structure there is called the eye spot. It's not image forming, but it is light sensing, okay? So I used to go collect horse manure for this lab, and we would culture that to look for the various protists in that. And if I keep the cultures in the dark, Euglena they don't produce the pigments. They will be heterotrophic because there's no light, but they do have the ability to be phototrophic. That is, they can take and use sunlight to photosynthesis, okay? So that's why these are green, okay? The eye spot is used to detect light, so they can maneuver to the right place to get the right amount of light. Not too much, not too little, and probably, but I don't know for certain, they can probably actually sense the quality of the light, that is the wavelength, and they can probably move to a position within the water column that best matches their absorption spectrum or action spectrum for photosynthesis. I don't know that definitively, okay? Things to help you when you use this microscope. One thing that happens a lot is the aperture diaphragm is too far open. Remember, you can use the field diaphragm, position condenser, etc., and the aperture diaphragm up by the lens, and I probably erased it already, but the aperture diaphragm, which is up here, right underneath the stage, but closer to the lens, that can be open and closed. So a lot of times what happens is it's just too far open, and as a result, you can't see this because you haven't introduced that contrast, okay? Now, you are losing some resolution, but I really can't make much out here. This is this, just close down a bit. I see more contrast. And you'll see cytoplasmic streaming in this amoeba as well. And you've seen these before, like these blobs, and you've probably seen Gary Larson Farsight cartoons where they're like, oh my god, the cover slip, right? It looks like they're being squashed. Keep in mind, there is a column of liquid there, but because the amoeba are very thick, the column of liquid would not be thick enough for that amoeba, so indeed it would be squashed if we didn't take steps to prevent that. So for some of the slides, we're going to have you add some grains of sand. That's why I said sometimes amoeba join as grains of sand. Then you'll add your drop with the amoeba solution. Take from the bottom of the culture, because amoeba tend to be bottom dwellers, so you have better luck finding them. Then you have your sand there, add your drop, put your cover slip on. That cover slip will not be that close to the slide because the grains of sand which allow the amoeba to move around. If you're using the 4x objective, the entire column of liquid will be in focus. You can start on that cover slip. Cover slip will be bigger than the field of view, so I'm going to draw the cover slip from looking down. There's the slide, there's the cover slip. It's not circular, we're using square ones. And then you position the field of view here. In one corner, you scan this way, you move it slightly down, scan. You can scan the entire cover slip in 30 seconds. Take and focus first on the edge of the cover slip, because you're not going to know where the specimens are. So if you focus on the edge of the cover slip, if you are in focus at the edge of the cover slip, at 4x objective, the entire column of liquid will be in focus. So if you get the edge of the cover slip in focus, the column of liquid will be in focus. So when you scan back and forth, everything should be in focus. You should be able to see any amoeba. If you scan this in 30 seconds, there are no amoeba, move on, make a new slide, okay? You can spend 10 minutes, but you're not going to find them if you didn't find them then, okay? Is that clear? A lot of people like to start with higher power because, hey, after all, you're going to use the higher power. But the problem there is, if you start with the higher power, like the 10x objective, your column, your depth of focus may be such that this entire column isn't in focus. So you may scan the entire film of water, but you're nowhere near where the amoeba are actually at. You may have scanned this layer of water in that column and never found your specimen. If you use the 4x objective, the entire column is in focus. Does that make sense? All right? That's that depth of focus that we keep talking about. Any questions? All right, let's move on. Here's the cheek. Take a toothpick. We have autoclaved the toothpicks. They are sterile. We and your fellow classmates would appreciate if you go in there with forceps and grab a toothpick, then take that toothpick, scrape the inside of your cheek. You don't have to be forceful. Little scraping. Take your drop of methylene blue. Now it's in a isotonic solution. So what does that mean? The methylene blue is in an isotonic solution. So what does that mean? So it means the tonicity of that saline solution with the dye matches the tonicity of the cell. So water does move back and forth, but it's at the same rate. So I want to go over the terms hypertonic and hypotonic. You always have to have a reference point for this term. So solution A, let's say that's 1 osmol. And B is 0.5 osmol. In terms, A is hypertonic to B, or I can say the exact same thing if I say B is hypotonic to A. So notice there's always a reference point, okay? So it is impossible to say solution A is hypertonic unless you have a reference. You always have to be comparing two things when you talk about tonicity. Isotonic, A and B are the same tonicity. If A is hypertonic, it has more osmolarity. Is that clear? Okay. So anyway, the outside dye solution methylene blue is isotonic. So the dye is free to move in and out. It preferentially binds to DNA. So what organelles would be stained? Nucleus? Mitochondria? Anything else? Chloroplast? Yes, no? Hopefully not in your cheek cell, right? Might be spinach, but that's not your cheek cell, okay? We'll do a lab in 2 weeks, the enzyme lab, where we'll talk about that biofilm and the oral debris on your teeth, because we'll actually have you spit in a container, and then we're going to have you spin it, and you'll see a nice big thick pellet of white stuff. You just call it. Anyway, we'll do that later. Here's those cheek cells. Now, here's the nucleus. Sometimes you can see nice, clear portions within the nucleolus. Within the nucleus. Give the answer away. What's that clear? What are the clear areas within the nucleus? Nucleolus. You can have more than one. The reason why it is clear, which is in contrast to almost every image you ever see from the nucleolus, usually the nucleolus is nicely darkly staining. Because the dye is specific for DNA, as it turns out, if you look at the nucleus, so I'm not going to draw it accurately, but if here's the nucleus here, I'm just going to draw it like this, no pores, no double membrane, none of that stuff for now. Within here are the chromosomes, eukaryote, and we'll be talking about the ribosomal. We've talked about ribosomes already. They're made up of RNA and protein. And as a result, those RNA genes are in specific locations. And we used to think, and we were learning a lot, we used to think these chromosomes are just randomly within this nucleus. In fact, they associate in specific patterns, and as a result, these ribosomal RNA genes are very close to one another, even though they may be on different chromosomes, different ribosomal RNA genes, and we make lots and lots and lots and lots and lots of ribosomal RNA. In fact, we make so much of that ribosomal RNA that there's not much DNA compared to the rest of the nucleus. So sometimes we literally see clear spots because there's not much DNA compared to the rest of the nucleus, okay? But all the figures always show the nucleus darkly stained because of the dye technique that's used. So depending on your dye technique will change what you see, okay? So anyway, with respect to the plant cells, we will have cell walls here, so we'll definitely be able to see that, okay? There's going to be the chloroplasts will be easy to see. The plant cells that we're looking at LED are big, so we'll see streaming. The Nightella will see streaming. We'll see this chloroplast moving, okay? Anyway, this is what the nucleus is going to look. Sorry, not Nightella, not Elodea. Yes, Elodea. The Elodea is going to look like this. Here's those chloroplasts. Now, this was precisely focused. You can actually see the nucleus here, the nucleolus there. I don't think any of you will see that. It's very unusual to find that. Just happen to be an excellent prep here, okay? We see lots of chloroplasts. Now, remember, we are optically sectioning the cell. So let's give an analogy that might help. I don't know basketball, but anyway, Michael Jordan, I know he doesn't. Kobe Bryant, he's still playing, right? Okay, so I have gone on to eBay and I bid on a Kobe Bryant basketball signed. He played with the national champions or whatever, okay? Really expensive. So, do you think they're just going to put it in a Safeway bag, address it, and ship it to me? I hope not. They're going to put it in a box. They're going to add packing peanuts. They're going to put that basketball in there. They're going to put packing peanuts around it. They're going to put packing peanuts on top. Wrap up the box. Put another box. Put another box. I hope. Okay. But the bottom line is, if we can optically section this, we can see anywhere within that box. Let's say I have to go to custom, the X-ray, whatever. So what we can do is on this plant cell, since it's transparent, the cell wall is thin enough that we can see light through it. We could look at the very top of that box, and we would see the cardboard. But if we went further, you'd see the packing peanuts, right? Well, if it was a plant cell, we could be at the top of the cell here. You'd see just the cell wall that corresponds to the cardboard. If we go further in, we would see the chloroplast, right? But remember, the plant cell has a large central vacuole, just like that basketball. It takes up a huge volume of that internal space, just like the central vacuole. So in the middle, if I'm in the middle of that cell, I would see the chloroplast around the perimeter, but not in the center. So what you can do is, I'll try to draw this on the board, and I know I'm not a good artist, but if this is that cell like this, and there's that large central vacuole, the nucleus, the chloroplast, my green chalk has disappeared, so pretend this is green, can be around like this. So if I'm at the very top, I would see something like this. Remember, we're looking down with the microscope, but our optical sections are in the vertical distance like this. So if I'm at the top, I'd see this and maybe just cell wall, so I don't see any chloroplast. When I, at the very next position there, again, I see the cell wall outline, so it's the same dimensions, but now I see all the chloroplast at the top there. It's like that layer underneath the box where I see all the packing peanuts. So I'd see all these chloroplasts like this, distributed throughout. But if I'm at the center point here, I still see the same perimeter, but now I have the central vacuole in the middle, and I got the chloroplast distributed around the perimeter. Just like if I'm in the center of that box with the basketball and packing peanuts. So what you do with the microscope is, at high power, thin section, gather that information in your brain, go to another position, gather information, another, and then you try to recreate the three dimensions. Okay? They do this with CAT scans, where they take the information at one plane, they move the body over the scanner, take another information, they piece those layers together to recreate the three-dimensional image. You do the same thing with optical, okay? Just keep in mind that I've drawn it from the side, and if we were, if I was really truly artistic, I'd make this three-dimensional, but we're looking down from the above. And when we move the stage, we always have that fixed position of focus, so as we move the stage up, now I can do this by moving the board, so there's that's in focus, move it up, that's in focus, move it up, that's in focus, move it up, that's in focus, right? So I'm gathering the information from each of those optical sections and then putting them together, okay? All right, so let's just go through a couple more things here. So that's sort of the key thing you want to be doing with this lab, is making those optical sections and recreating that three-dimensional image. If there's time, we're going to have you look at diatoms, and the purpose of this is trying to introduce some statistics, some analysis. So you look at the shape, there are some that are tri-cornered like this and some that do not have the three corners. So we're going to look at various cultures and try to figure out ratios of those. Now, for lab, this is where we start doing the Vibrio work. So we're going to take that seawater, we're going to have a plate, SWC plate. It's a Petri plate, and from the side it looks like this. So from the side, it looks like this, with a lid, it's plastic, and an agar surface here. So there's agar there, kind of like jello. What we need you to do is take the lid off. The best way to do this with sterile technique is take the lid and don't set it aside because the surface is contaminated. Rock the lid up a little bit, squirt your 100 microliters onto there from the seawater using sterile technique to the tip, and then you spread that. So the geoscience will demonstrate this in small groups. We are working with open flame, so please tie back long hair, be careful. But the bottom line is, from the top view, we see a circle like this. So what you have to do is lift the lid up, add your 100 microliters to this solution to the surface here, then you want to uniformly spread that out, okay? Now I didn't bring the spreader here, but it's basically a rod that you're going to dip in alcohol and flame sterilize it, and if you have that circular plate, what you're going to do is take that rod, go back and forth like this to spread those cells out, rotate the plate about maybe 120 degrees, sterilize it again, spread it out, do it again. That way, hopefully you'll get a nice uniform spread of that solution, so that next week when you see this, hopefully we're going to have nice isolated colonies like this, and hopefully some of them will be bioluminescent, and we'll see that in lab, and that's the one we're going to use to freak out, okay? So that's the hope that as we proceed with this, okay? So anyway, so let's just run through this. That's that plating. Here's what it looks like next week when you see this. You're going to find colonies of different sizes. This is a fast grower, slow grower. The ones that are really slow growing, you never see them because you pull the plate out. Okay? So we're looking for the bioluminescent ones. You're going to pick from this using sterile loops in another week after this. You're going to use that loop to spread them out. The whole purpose of this is if you start with this on that loop, there's going to be tens of thousands of bacteria when you pick that colony. So when you spread this out, there's going to be lots of bacteria there. When you get a new loop, there's none on it because it's sterile. You're going to go in, cross this partially, pick up a bunch of bacteria, spread them out, distributing them, decreasing density. Next loop, cross this, you're going to pick up some bacteria, spread them out. Ideally, when we get to this point here, there's going to be isolated bacteria that when we see them next time, it's a colony starting from just one. Now we don't know if this is from just one, probably was, but to be safe, we're going to streak. We're going to streak again. Because what we're going to do, we're going to keep streaking this, so we're really sure that we have a colony from one starting bacterium. Then we're going to do PCR. Remember, DNA is double-stranded and anti-parallel. And we have some information about the background of the chromosomes here, okay? So these are bacteria, so the DNA is circular. Oh, don't know. I hate PowerPoint. Anyway, font that E should be there, but anyway, circular chromosome. Here's our target gene. That's what we want to amplify. That's what we want to make lots and lots of copies of, okay? So we have some information about what the background should be like. It's sort of like, you go to a new city, you want to buy a house, you know the parameters of the neighborhood you want. Nice schools, low crime, etc. That allows you to target the location. In this case, we target location because we know the flanking information here. We don't know the exact information within here. We're going to sequence it. But we know flanking information, so we can generate what's called primers. That is, we know this sequence there and this so we can design complementary DNA that's single-stranded called primers. What we'll do is we'll separate the two strands. How are they held together, the two strands? Hydrogen bonds are covalent. Hydrogen bonds. So we just heat it up. That will denature the DNA. Separate the two strands. And remember, they're antiparallel. Separate the two strands. The primers anneal at specific spots. They're antiparallel. So this one would anneal to that strand. This primer would anneal to that. And then we'll allow it to extend using a DNA polymerase. And we'll basically anneal primers, extend, make new ones, separate those two double strands, do 4 single strands. Primers will anneal again. So basically double the amount of DNA from 1 double strand to 2 double strands to 4 to 8, 16, etc. Okay? We'll quantitate that on the gel. First, we got to make sure we have the band. Is it of the size we expected? How much is there? Once we quantitate it, we're going to see bands. We can say there's that much. Once we know the quantity, we can set up reactions for sequencing. We'll send the DNA out. So I still have about 5 minutes, so if we can maybe just keep a little bit quiet, please. So we're going to send it out for sequencing. We'll do this more. I'm trying to give you this because once we start doing the enzyme lab in the other labs, there just isn't much time to do this. So we will extend this DNA. Here's the primer. We'll extend it using DNA polymerase. We have to have the building blocks. And we'll extend it in the PCR reaction. When we do sequencing, we'll have things that are called terminators, which will stop synthesis. So when we send it out for sequencing, we're going to get data back that looks like this. That is of the strand being made. So we will know exactly the strand being made, which, of course, you know what the complementary strand was, right? So we will generate the sequence. You'll put it in the database. Once we figure out the sequence, you'll put it in the database. That will come back and say, hey, it's probably this species. These are the number of bases that matched. You'll pick these, and then between the base groups in lab, hopefully we'll have enough diversity that we can generate trees that show the relationship of this student's bacterium versus this versus that in there, okay? So it allows us to use the computer to generate this sort of analysis, okay? And I do want to point out, just for those of you, there's no lecture on February 18th. That's a holiday. We do have lab though on Tuesday through Friday, so we'll be able to photosynthesis lab then, okay? Now, actually, I have just about 4 minutes, so what I'm going to do now is show you the PCR, how it works, okay? And I will post this on BSpace, and hopefully it will make sense. Okay, where did it go? Okay, so I will narrate it as we go through. So it's PCR called polymerase chain reaction. Discovered by Kerry Mullis. He's a UC Berkeley graduate. Reagents. Template. That's our starting source. In this case, it's the DNA in the bacterial colony, right? So that's our template. We have primers. We're targeting 2 genes. So for the RNA polymerase gene, we have the primers that flank it. For the RPOB, the RNA polymerase gene, we have the 2 primers that flank it. So clear what the primers are. They flank either side, represent each of the 2 strands. Taq polymerase is the DNA polymerase. It just extends onto that primer. Using the information of the other base to know what should go in. We have to have the building blocks, and of course we always have to have buffers and enzymes and things like that. Okay. Remember, DNA is antiparallel. Okay. Synthesis will be 5 prime to 3 prime. The steps are denaturation, separating the 2 strands, annealing, allowing the primers to bind at specific locations, extension, extending that, and we'll do that a bunch of times. So let's just see how this works. If we start off with template here, it's the exact length of our target. So there's our template, denature. We have our primer 1. It anneals there. We need to have a second primer. It anneals there to the other strand. There, what we've got is we had our template. We had the 2 primers, anti-parallel, flanking this. We extend it. So primers come in. Extend. We now have 2 molecules identical to what we started with. Denatured again. We're going to have go through this. We now have 4 single strands. Our primers anneal again. We're going to get 4 double strands. We separate. 8 single strands. Primers anneal. We're going to get 8 templates, 8 double strands. So we're always doubling. So after 10 rounds, we go from 1 to about 1,024. After 20 rounds, we go to 1,000,000. After 30 rounds, we go to 1 billion. I'll do the math later for you. We're always doubling. 2 to the 10th is 1,024 or about 1,000. Okay. So and then next time, think about what happens when we have a different situation. This was where the primers were exactly at the ends of our target. But in reality, when we're doing our experiment, our gene is in the background of that huge chromosome. So the way this works will be slightly different in terms of what we get. So anyway, we'll do more on the PCR stuff as we go through. So enjoy lab.